Page number 186, chapter number 10, Intermediate Secondary Botany. Okay. RNA world. From the foregoing discussion, an intermediate question becomes evident, which is the first genetic material. It shall be discussed in detail elsewhere under the chemical evolution. But briefly, we shall highlight some of the facts. So, RNA was the first genetic material. వామో ఇది ఇంపార్టెంట్ అంతా తెలియదు ఆర్ఎన్ఏ డీఎన్ఏ అయినా ఫస్ట్ జెనెటిక్ మెటీరియల్ అంటే ఆర్ఎన్ఏ ఇస్ జెనెటిక్ మెటీరియల్ అంట ఓకే RNA was the first genetic material. There is now enough evidence to suggest that essential life processes essential life processes such as metabolism, translation, and spilling, etc. evolved around RNA. RNA used to act as a genetic material. RNA used to act as a genetic material as well as a catalyst. So there are some important biochemical reactions into living systems that are catalyzed by RNA. So there are some important biochemical reactions in living system that are catalyzed by RNA catalyses and not by protein enzymes. So catalytic RNA or RNA enzymes are known as the riboenzymes. Okay, ribozymes. Okay, ribozymes. Catalytic RNA or RNA enzymes are known as the ribozymes. But RNA, being a catalyst, was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modifications that made it more stable. DNA. Okay. Therefore, DNA has evolved from RNA with the chemical modifications that made it more stable DNA. Okay, more stable DNA, being double stranded, and that it more stable DNA, being double stranded, having complementary strands, further resistant changes by evolving a process of repair. Next, replication గురించి. Replication. While proposing a double helical structure of DNA, Watson and Crick had immediately proposed a scheme for replication of DNA. To quote their original statement that is as follows. So it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. So Watson and Crick 1953. So it has not escaped our notice that the specific pairing we have postulated immediately suggested a possible copying mechanism for the genetic material. So Watson and Crick 1953. The scheme suggested that the two strands would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have a one parental and one newly synthesized strand. So this scheme was termed as the semi-conservative DNA replication. Figure 10.8 లో ఉంది చూడండి. Okay. Next, the experimental proof. దేనికోసం చూద్దాం ఒకటి. Okay. It is now proven that DNA replicates semi-conservatively. So it is now proven that DNA replicates semi-conservatively. It was shown first in E. coli. Okay. It was shown first in E. coli and subsequently in higher organisms such as plants and human cells. Matt Howe Michelson and Franklin Stahel performed the following experiment in 1958. They grow E. coli in the medium containing NH4Cl15, and 15N is the heavy isotope of nitrogen. as the only nitrogen source for many generations. The results was that 15N అంటే 15N అనేది చెప్పాం కదా మనం is the heavy isotope of nitrogen అనమాట. The 15N was incorporated into newly synthesized RNA as well as other nitrogen containing components. This heavy DNA molecule could be distinguished. This heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride (CsCl) density gradient. Okay, the heavy DNA molecule could be distinguished from the normal DNA by centrifugation in a cesium chloride (CsCl) density gradient. Okay, density gradient. So note that N nitrogen is not an radioactive isotope. Note that N nitrogen is not a radioactive isotope, and it can be separated from N14 based on the densities only. Then they transferred the cells into the medium with normal NH4Cl14 and took samples at various definite time intervals as the cells multiplied, and extract the DNA that remained as a double-stranded helix. And the extracted the DNA that remained as a double-stranded helix. The various samples were separated independently on CsCl gradients to measure the density of DNA. So the results are shown in figure 10.9. Can you recall what centrifugation force is, and think why a molecule with high mass and density would sediment faster? Can you recall what centrifugal force is, and think why a molecule with high mass and high density would cement faster? Third one name that's the DNA. ఈ విధంగా the DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium. So that is after 20 minutes. Equally divides in 20. That is after 20 minutes the equally divides in 20 minutes. Had a చూడండి ఇక్కడ ఏమంటే the transfer from N15 medium to N14 medium had a hybrid and intermediate density. Okay, density ఇక్కడ full stop ఉంది. Next, DNA extracted from the culture after another generation, that is after 40 minutes. Okay, DNA extracted from the culture after another generation, that is after 40 minutes. Second generation. Okay, DNA extracted from the culture after another generation was composed of equal amount of this hybrid DNA and of light DNA. Hybrid DNA and light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the proportion of DNA molecules with the light and hybrid densities? ఇంకా 20 నిమిషాలు ఎక్కువ పెంచుంటే ఏమవుతుంది అని అడుగుతుండే అక్కడ. సో very similar experiments involving the use of radioactive thymidine to detect the distribution of newly synthesized DNA in the chromosomes was performed on Vicia faba. And faba beans by Tyler and his colleagues in 1958. The experiments proved that the DNA in chromosomes also replicate semi-conservatively. semi-conservatively అని చెప్తాను. The machinery and the enzymes. In the living cells such as E. coli, the process of replication requires a set of catalytic enzymes. The main enzyme is referred to as a DNA-dependent DNA polymerase. Since it uses a DNA template to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. In a very short time. E. coli that has only 4.6 into 10 to the power of 6 base pairs. Compare it with humans whose diploid content is 6.6 into 10 to the power of 9 base pairs. Okay, so E. coli that has only 4 into 4.6 into 10 to the power of 6 base pairs completes the process of replication within 38 minutes. This means the average rate of polymerization has to be approximately 2000 base pairs per second. That means the average rate of polymerization has to be approximately 2000 base pairs per second. So not only do these polymerases have to be fast, but also have to catalyze the reaction with a high degree of accuracy. Not only do these polymerases have to be fast, so polymerase ఉండడం వల్ల fast మాత్రమే కాదు, but they also have to catalyze the reaction with a high degree of accuracy. ఈ పాలిమరేస్ అనేటివి రియాక్షన్ ని ఫాస్ట్ చేయడమే కాకుండా మరియు రియాక్షన్ ని క్యాటలైజ్ అంటే హై డిగ్రీ ఆఫ్ అక్యురసీతో కూడా చేస్తాయన్నమాట ఈ పాలిమరేజ్ ఉండడం వల్ల. Any mistake during replication would result in mutation. Any mistake during replication would result in mutation. అవును. Furthermore, in terms of energy, replication is a very expensive process. Deoxyribonucleoside triphosphate serve as a dual purpose. Deoxyribonucleic acid triphosphate serve as a dual purpose. In addition to act as substrate, they provide energy for polymerization reactions. The two terminal phosphates in a deoxynucleoside triphosphate are higher energy phosphates, as in case of ATP. Okay. In addition to DNA dependent DNA ligases, in addition to DNA dependent DNA polymerases, many additional enzymes are required to complete the process of replication. In addition to DNA dependent DNA ligase, దాంతో పాటు కాకుండా many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, since the two strands of DNA cannot be separated throughout the entire length in one stretch, so due to very high energy requirement. గుర్తుపెట్టుకోండి ఇక్కడ, ఫర్ లాంగ్ డిఎన్ఏ మాలిక్యూల్స్ సిన్స్ ద టూ స్టాండ్స్ ఆఫ్ డిఎన్ఏ కెనాట్ బి సపరేట్ త్రూఅవుట్ ది ఎంటైర్ లెంత్ ఇన్ వన్ స్టిచ్. ఎందుకు స్ట్రెచ్ అవ్వదంటే, ఎందుకు విడిపోదు మొత్తం అంటే, డ్యూ టు వెరీ హై ఎనర్జీ రిక్వైర్మెంట్, ఎక్కువ ఎనర్జీ కావాలి రెప్లికేట్ కావాలంటే. సో ద రెప్లికేషన్ అకర్స్ విత్ ఇన్ ఏ షార్ట్ ఓపెనింగ్ ఆఫ్ డిఎన్ఏ హెలిక్స్. సో referred to as replication fork. The DNA dependent DNA polymerases catalyze the polymerization only in one direction, that is 5 to 3 direction, 5 prime to 3 prime direction, 5 prime to 3 prime direction. ఇప్పుడు గుర్తొచ్చింది, 5 prime అంటారు, then 3 prime directions అంటారు. So this creates some additional complications at the replicating fork. Consequently, on one stand, the template with polarity 3 prime to 5 prime, the replication is continuous. This stand is known as the leading stand, while the other it is discontinuous. So while the other అంటే ఇక్కడ template with polarity 5 prime to 3 prime direction అన్నమాట. So it is a discontinuous. It is the lagging stand. The discontinuously synthesized fragments known as the Okaji. Okay. Okazaki fragments. Okay. Okazaki fragments. The discontinuously synthesized fragments known as the Okazaki fragments and later joined by the enzyme DNA ligase. And later joined by కలుపుతాయి మళ్ళీ మొత్తం later enzymed by the DNA ligase, figure 10.10. Okay. The DNA polymerase on their own cannot initiate the process of replication. A small stitch of RNA called a primer is required for initiation. గుర్తుపెట్టుకోండి, DNA polymerase అనేది తనంతట తానే రెప్లికేషన్ స్టార్ట్ చేయదు. A small stretch of RNA అంటే called the primer is required for initiation. So it is used as a anchor for the addition of new DNA molecules, nucleotides. It is used as a anchor for the addition of new DNA nucleotides. The primer is removed later. Also, the application does not initiate randomly at any place in the DNA. Also, the replication does not initiate randomly at any place in the DNA. Rather, this is a definite reason in E. coli DNA where the replication. Rather, there is a definite reason in E. coli DNA where the replication originates. Such a reason is termed as the origin of replication. Okay, or ORI. Okay, మనతో మాట్లాడుకున్నాం ORI. O అంటే ఆరిజిన్, R అంటే రెప్లికేషన్, I అంటే origin, ORN. So it is because of the requirement of the origin of replication. It is because of the requirement of this origin of requirement. Origin of requirement అనేది ఒక పదం. So that a piece of DNA, if needed to be propagated during recombinant DNA procedures. That a piece of DNA, if needed to be propagated during recombinant DNA procedures, requires a vector. The vectors provide the origin of replication. The vector provides the origin of replication. Further, not every details of replication is understood well. Further, not every detail of replication is understood well. Further, every details of replication is not understood well. In eukaryotics, the replication of DNA takes place at the S phase of the cell. In eukaryotics, the replication of DNA takes place in the F phase of the cell. The replication of DNA and cell division cycle should be highly coordinated. A failure in cell division after DNA replication results in polyploids, a chromosomal anomaly, So you will learn the detailed nature of the origin anomaly. You will learn the detailed nature of the origin and the process occurring at this site. You will learn the detailed nature of the origin and the process occurring at this site in higher classes. Okay. Next to page number 190 లో transcription గురించి. The process of copying genetic information from one strand of DNA into RNA is called the transcription. Here also the principle of complementarity. Here also the principle of complementarity governs the process of transcription, except that the adenosine now base pair with except that the adenosine now base pair with the uracil instead of thymine. However, unlike the process of replication in which once set in the total DNA of the organism get duplicated. So unlike in the process of the replication in which once set in, the total DNA of the organisms get duplicated. In transcription, only a segment of DNA and only one of the strands is copied into RNA. In transcription, only a segment of DNA and only one of the strands is copied into RNA. So this necessitates defining the boundaries. That would demarcate the region and the strand of DNA that would be transcribed. Okay, that would be transcribed. Okay, why both strands are not copied during transcription has a simple answer. So why both the strands are not copied during the transcription has a simple answer. Okay, so first, if the both strands act as a template, they would code for RNA molecules with different sequences. So remember, complementarity does not mean being identical. And in turn, if they code for proteins, the sequence of amino acids in the protein would be different. and in turn if they code for proteins, the sequence of amino acids in the protein would be different. Hence one segment of DNA would be coded one segment of DNA hence one segment of DNA would be coded for different proteins, and this would complicate the genetic information transfer the machinery, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules. Second, the two RNA molecules if produced simultaneously. Second, the two RNA molecules if produced simultaneously would be complementary to each other. Hence would form a double-stranded RNA. So this would prevent the RNA from being translated into proteins, and the exercise of the this would prevent the RNA from being translated into protein, and the exercise of transcription would become a futile one, and the exercise of transcription would become a futile one. Okay. Transcription unit in a DNA is defined primarily by the three regions in the DNA. A transcription unit in DNA is defined primarily by three regions in the DNA. One is promoter, second structural gene, థర్డ్ వన్ ఏమో టర్మినేటర్. ప్రమోటర్, స్ట్రక్చరల్ జీన్, టర్మినేటర్. ఒకటేమో ప్రమోటర్, రెండోదేమో స్ట్రక్చరల్ జీన్, మూడోదేమో టర్మినేటర్. There is a convention in defining two strands of DNA in the structural gene of the transcription unit. Since the two strands have opposite polarity. Because ఒకటే. So because the two strands have opposite polarity and the DNA dependent RNA polymerase, ఇక్కడ చూడండి వెరీ ఇంపార్టెంట్, ఇక్కడ DNA dependent RNA polymerase or transcriptase also catalyze the polymerization in only one direction, that is 5' to 3' direction. The strand that has the polarity 3' to 5' act as a template and is also referred to as the template strand. The other strand which has the polarity 5' to 3' direction and the sequence same as the RNA except thymine at the place of the uracil. So is replaced during transcription. So strangely this strand does not code for anything. For anything. ఏ స్టాండ్ అయితే ఏ కోడింగ్ కి గురి కాలేదు ఆ స్టాండ్, దిస్ స్టాండ్ ఈజ్ రెఫర్ టు యాజ్ ది కోడింగ్ స్టాండ్. స్ట్రేంజ్లీ దిస్ స్టాండ్ ఈజ్ రెఫర్ టు యాజ్ ది కోడింగ్ స్టాండ్. ఎందుకు? ఆ స్టాండ్ అనేది డస్ నాట్ కోడ్ ఫర్ ఎనీథింగ్, ఎటువంటి కోడ్ అనేది దానికి పొందలేదన్నమాట. ఆల్ పాయింట్స్ వైల్ డిఫైనింగ్ ఏ ట్రాన్స్క్రిప్షన్ యూనిట్ ఆర్ మేడ్ విత్ రెఫరెన్స్ టు ది కోడింగ్ స్టాండ్. All points while defining a transcription unit are made with reference to the coding stand. To explain the point, a hypothetical sequence. From the transcription unit is represented below. To explain the point, a hypothetical sequence from the transcription unit is represented below. Okay. చూడండి ఇక్కడ టెంప్లెట్ స్టాండ్ ఇచ్చాడు, కోడింగ్ స్టాండ్ ఇచ్చాడు, గుర్తుపెట్టుకోండి. టెంప్లెట్ స్టాండ్ ఏమో 3' to 5' ఉంది, కోడింగ్ స్టాండ్ ఏమో 5' to 3' ఉంది, రివర్స్ అనమాట ఇక్కడ. ఓకే. కెన్ యూ నౌ రైట్ ద సీక్వెన్స్ ఆఫ్ RNA ట్రాన్స్క్రైబ్డ్ ఫ్రమ్ ద అబోవ్ DNA? కెన్ యూ రైట్ ద సీక్వెన్స్ ఆఫ్ RNA ట్రాన్స్క్రైబ్డ్ ఫ్రమ్ ద అబోవ్ DNA? The promoter and terminator flank the structural gene in the transcription unit. The promoter and the terminator flank the structural gene in the transcription unit. The promoter is said to be located towards the 5' end upstream of the structural gene. The reference is made with respect to the polarity of coding stand. The reference is made with respect to the polarity of coding stand. It is a DNA sequence. Okay, it is a DNA sequence that provides the binding site for RNA. Polymerase, and it is the presence of the promoter in the transcription unit. That also defines the template and coding stand. By switching its position with that, By switching its position with that of the terminator, the definition of coding and template stands can be reserved. The definition of a coding stand and template stands can be reserved. The terminator is located towards the 3' end downstream of the coding stand and usually defines the end of the process of transcription. The terminator is located towards the third 3 end downstream of the coding stand and it usually defines the end of the process of the transcription. Figure 10.11. So there are additional regulatory sequences. There are additional regulatory sequences that may be presented further upstream and downstream of the promoter. So there are additional regulatory sequences that may be present further upstream and downstream of the promoter. Some of the properties of these okay some of the properties of these some of the properties of these some of the properties of these sequences shall be discussed while dealing with the regulation of the gene expression. నెక్స్ట్ వచ్చేసి టైటిల్ ట్రాన్స్క్రిప్షన్ యూనిట్ అండ్ ద జీన్ ఓకే. A gene is defined as the structural functional unit. A gene is defined as the functional unit of inheritance. Though there is no ambiguity that the genes are located on the DNA. So it is difficult to literally define a gene in terms of DNA sequence. It is too difficult to literally define a gene in terms of DNA sequence. The DNA sequence coding for tRNA transfer tRNA or rRNA molecule also define a gene. However, by defining a cistone as a segment of DNA coding for a polypeptide, the structural gene in a transcription unit can be said to be monocystonic. Mostly eukaryotics are polycystic. Monocystonic. Mostly in eukaryotics are polycystonic. Mostly in bacteria or prokaryotics. In eukaryotics, the monocystonic structural genes have interrupted coding sequences. The genes in eukaryotics are split. అంటే ఏంటంటే the genes in eukaryotics are split అని అర్థం. The coding sequences or expressed sequences are defined as exons. Exons are said to be those sequences that appear in mature or proceed RNA. Exons are said to be those sequences that appear in mature or proceed RNA. The exons are interrupted by introns. Introns are intervening sequences. Introns and intervening sequences do not appear in mature or processed RNA. The split gene arrangement further complicates the definition of the gene in terms of DNA segment. Inheritance of a character is also affected by the promoter and regulator sequences of the structural gene. Inheritance of character is also affected by promoter to or regulating sequence of the structural gene to. Hence, sometimes the regulatory sequences are loosely defined as regulatory sequences. Genes, even though these sequences do not code for any RNA or DNA. So, even though these sequences do not code for any RNA or protein. Okay. Types of RNA and the process of transcription. In bacteria, there are three major types of RNAs. mRNA అంటే messenger RNA, tRNA అంటే transfer RNA, rRNA ribosomal RNA. All three RNAs are needed to synthesize a protein in a cell. All three RNAs are needed to synthesize a protein in a cell. The mRNA, messenger RNA, provides the template, and the tRNA reads the genetic code, bringing amino acids, brings the amino acids. Okay, and rRNA plays a structural and a characteristic role during translation. So there is a single DNA dependent RNA polymerase. There is a single DNA there is a single DNA dependent RNA polymerase that catalyzes the transcription of all types of RNAs in bacteria. There is a single DNA dependent RNA polymerase that catalyzes the transcription of all types of RNA in bacteria. RNA polymerase bind to the promoter. RNA polymerase bind to the promoter and initiates the transcription. RNA polymerase bind to the promoter and initiates the transcription initiation. Okay, it uses the nucleoside triphosphate as substrate. It uses the nucleoside triphosphate and దాన్ని substrate గా యూజ్ చేస్తుంది, and polymerases in a template dependent fashion following the rule of complementarity. And polymerases in a template dependent fashion. చూడండి, ఇక్కడ ఇది ఏం చెప్తుందంటే ఇట్ యూజెస్ ది న్యూక్లియోసైడ్ ట్రైఫాస్ఫేట్ అనే దాన్ని సబ్స్ట్రేట్ గా యూజ్ చేస్తుంది, అండ్ పాలిమరైజెస్ ఇన్ ఏ టెంప్లేట్ డిపెండెంట్ ఫ్యాషన్ ఫాలోయింగ్ ద రూల్ ఆఫ్ కాంప్లిమెంటారిటీ. ఓకే, it somehow also facilitates the opening of the helix and continuous elongation. It somehow also facilitates the opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So all the RNA polymerase. The nascent RNA falls off. So also the RNA polymerase results in termination of transcription. This results in termination of the transcription. An intriguing question is that how is the RNA polymerase is able to catalyze all the three steps: initiation, elongation, termination. RNA polymerase is only capable of catalyzing the process of elongation. RNA polymerase is only capable of catalyzing the process of elongation. It associates transitionality. Okay, it associates transitionally with initiation factor. Alpha and termination factor beta to initiate and terminate the transcription respectively. Association with these factors alter the specificity of RNA polymerase to either initiate or terminate. Figure 10.12. In bacteria, since the mRNA does not require any processes to become active. And also since transcription and translation takes place in the same compartment, so there is no separation of cystole and nucleus in the bacteria. Many times the translation can begin much before the mRNA. Is fully transcribed. Okay. Consequently, the transcription and the translation can be coupled in bacteria. Okay. In eukaryotes, there are two additional complexities. There are at least three RNA polymerase in the nucleus, in addition to the RNA polymerase found in the organelles. So there is a clear cut division of labor. There is a clear-cut division of labor. The RNA polymerase 1 transcribes the rRNAs. RNA polymerase 1 transcribes the rRNAs 28S and 18S and 5.8S. The RNA polymerase 2 transcribes the precursor of mRNA. The RNA polymerase 2 transcribes the precursor of mRNA, the heterogeneous nuclear RNA. HN ante heterogeneous nuclear RNA. Whereas RNA polymerase 3 is responsible for transcription of tRNA, 5SR RNA and SN RNAs, small nuclear RNAs అన్నమాట. SN అంటే ఇక్కడ small nuclear RNAs అన్నమాట. The second complexity is that the primary transcription contains both exons and introns and are non-functional. Hence, they are subjected to a process called the spilling. Where the introns are removed and exons are joined in a defined order. Heterogeneous nuclear RNA undergoes additional processing called the capping and tailoring. In capping an unusual nucleotide, methyl gynosin triphosphate. చూడండి ఇక్కడ ఇన్ క్యాపింగ్ అండ్ అన్యూజువల్ న్యూక్లియోటైడ్ అంటే ఏంది? అన్యూజువల్ న్యూక్లియోటైడ్ పేరు ఏంటంటే మిథైల్ గ్వైనోసిన్ ట్రైఫాస్ఫేట్ ఈజ్ యాడెడ్ టు ద 5' ఎండ్ ఆఫ్ హెటరోజెనస్ న్యూక్లియర్ ఆర్ఎన్ఏ. ఇన్ టైలరింగ్ ఎడినిలేట్ రెసిడ్యూస్. అనే దాంట్లో ఎడినిలేట్ రెసిడ్యూస్ 200 to 300 are added to at 3' end in a template dependent manner. So it is the fully processed heterogeneous nuclear RNA, now called the mRNA. That is transported out of the nucleus for translation. So now called the mRNA. ఇప్పుడు ఈ mRNA అనేది ట్రాన్స్పోర్టెడ్ అవుట్ ఆఫ్ ది న్యూక్లియస్ ఫర్ ట్రాన్స్లేషన్ పర్పస్ కోసం బయటికి వెళ్ళిపోతుంది అనమాట. అది సో ఫిగర్ 10.13లో చూస్తే మనకు అర్థమవుతుందని చెప్తాను. ఓకే, the significance of such complexity is now beginning to be understood. The split gene arrangements, ఇదంతా ఒక పదము. The split gene arrangements represent probably an incident feature of genome. The presence of introns is reminiscent. Okay, the presence of introns is reminiscent of antiquity. The presence of